Today the topic of our discussion is blue-white screening. What is blue-white screening? So blue-white screening is actually a laboratory or experimental technique which helps to distinguish between the recombinant plasmid containing bacteria from the non-recombinant plasmid containing bacteria. And the basis of this differentiation in the blue-white screening is the activity of beta-galactosidase enzyme. We will learn about this entire process, principle, and the concepts of every component in this video. So when we perform a blue-white screening experiment, we get to see a plate like this. You can see here are some colonies which are blue in color and some colonies which are white in color. The reason for these colonies, blue and white, we will learn separately in this video. Blue colonies. So for blue colonies, we will see that at first this is the normal E. coli cell. A normal E. coli cell containing the plasmid on the left hand side and the bacterial chromosome. If we zoom into the plasmid of this normal E. coli cell, we have this blue region which is the LAC-Z region. This LAC-Z region codes for beta-galactosidase enzyme. Now this beta-galactosidase enzyme converts lactose into galactose and glucose. Beta-galactosidase enzyme also can convert X-gal into a blue colored compound, that is 5,5-dibromo 4,4-dichloro indigo. Now X-gal is actually an analog of lactose. In the media that we use in the blue-white screening experiment, it has agar, X-gal and IPDG. X-gal is actually a chromogenic substrate. This compound converts into a blue colored compound which is helpful for identification. So you see the plate, this plate containing the blue colored colonies, and these colonies are formed by the bacteria that has a intact plasmid. That means their LAC-Z region is totally functional and it is not disrupted. That means it is not recombinant. So it's a non-recombinant bacteria, and these bacteria will give the blue colonies. Now the white colonies. So again we have a normal bacterial cell, normal E. coli bacterial cell, and it has the plasmid, and this plasmid we cut open the LAC-Z region by cutting with the restriction endonucleases or restriction enzymes, same thing. So we cut open this LAC-Z part, and this part can means the restriction endonucleases, it can be it can cut a blunt end or it can cut the sticky ends. Depending on the type of the restriction endonuclease you are using, you can get that specific cut. And similar, make sure that you cut the gene of interest that you are using. This gene of interest, this black segment is here the gene of interest. You cut the gene of interest with the same restriction endonuclease so that you can ligate it using DNA ligase and put it in this cut region. So you can see in the cut region we have put the black segment, that is the gene of our interest in this experiment, and we disrupted the LAC-Z region, and thus this plasmid is now considered as a recombinant plasmid. And since this plasmid is now disrupted, it is not functional anymore. LAC-Z gene is not functional anymore. Okay. So we transform the E. coli cells with this recombinant plasmid, and now this plasmid will not code for the beta galactosidase enzyme. As a result of this, the reaction is also not done. That is, the X-gal to the blue colored compound formation is also not done. And as a result, we will get these white colonies. So the white colonies are indication of the fact that the plasmid that has been incorporated in the bacteria is recombinant in nature. So they will make the white colonies because the X-gal is not converted into the blue colored compound because the beta galactosidase enzyme is not formed. Now there are chances that you can get a false blue colony. Let's see the reason for that. So you have a normal E. coli cell again. Earlier, you see, we considered only the plasmid sections. But a bacteria also contains a bacterial chromosome, which contains bacterial DNA, and that codes for the lac operon. So this is the lac operon region containing the capsid, promoter, operator, lac Z, lac Y, and lac A. This set is called the lac operon. Now, here the lac Z, it can code for the beta galactosidase and it can convert the X-gal into the blue colored compound, and we can get the blue colonies here. So you see, if you see the original bacterial cell, it contains the recombinant plasmid here. And we are supposed to get the white colonies. But instead of that, we are getting the blue colonies here. Why? Because this bacterial chromosome, it has the lac operon inside it, which is coding for the beta galactosidase enzyme, and we are getting the blue colonies. Now this is a false signal, and so this problem has been resolved by alpha complementation. So in laboratory, we usually use the E. coli cells, which are usually the DH5 alpha E. coli cells we use, and it has we incorporate the cell with a modified plasmid. This modified plasmid has the lac Z region, which codes for only certain subunits of the beta galactosidase enzyme, and this chromosome here we perform a deletion mutation in the lac M delta 15 deletion mutation we perform in this chromosome, and in the bacteria, means in the bacterial DNA we actually create this mutation, and as a result of this mutation, it is also not capable of producing a functional beta galactosidase enzyme by its own. So this plasmid containing the modified lac Z region will code for the alpha subunits of the beta galactosidase enzyme, and the parts and the modified parts of the bacterial this DNA, it will code for the omega subunits. Now these two subunits will merge together to form the functional beta galactosidase enzyme. Now this functional beta galactosidase enzyme can convert the X-gal into the blue colored complex, and we will get the blue colonies here. So this is indication of the fact that our lac Z region, although it is modified, but it is not recombinant. It does not have our gene of interest incorporated in the lac Z region. So it is not a recombinant. So we are getting means we can relate now that the blue colonies means the ones that do not contain the recombinant plasmid in the cell. And if we have a recombinant plasmid, so you see this black segment again in the middle of the lac Z region. So our lac Z region, the modified lac Z region, it is becoming non-functional due to the incorporation of the gene of interest, and as a result no subunits are produced, but we can the bacterial DNA, it is functional in nature, and it will code for the omega subunits. And as a result, we will get a non-functional beta-galactosidase, because both alpha and omega, these two subunits are needed for the functional enzyme. But since we are only getting the omega subunits, the enzyme is not functional, and as a result, the X-gal is not converted to the blue colored compound, and we are getting the white colonies. So now this alpha complementation, this concept has like resolved the earlier problem of false signals. Now the role of IPDG. So earlier I mentioned that in the media that we use in the blue-white screening experiment, we are using the agar, X-gal and IPDG. So X-gal, now we know X-gal is the chromogenic substrate which gives the blue colored compound and white colored compound, and which helps us to like distinguish between the recombinant, non-recombinant bacteria. Now what is the role of IPDG? So IPDG is actually an analog of allolactose, and you see this is the lac operon region, and when the media has no lactose in it, so the repressor, it binds the operator region, and as a result of this repressor binding, the transcription is inhibited. That means the LAC-Z region will not code for the beta-galactosidase enzyme when the lactose is absent in the media. Now, if lactose is present in the media and the repressor is initially bound to the operator region, now this lactose is taken up by the cell and it is converted to allolactose, and this particularly this allolactose, and this allolactose actually IPDG is actually an analog, like it has similar function like the allolactose. So IPDG is an analog of allolactose, and this compound specifically binds to the repressor, and upon binding to the repressor, it removes the repressor from the operator region, and next the cap binds to the cap site, and the RNA polymerase also binds to the promoter region, and the transcription of the LAC-Z, LAC-Y, LAC-K regions are initiated. So we can say that the IPDG, this compound is actually an inducer of the transcription. It induces the gene expression of the LAC-Z region, and thus the beta-galactosidase enzyme is like the production of beta-galactosidase enzyme is induced in the bacterial, from the bacterial DNA. So this is the role of IPDG, and this is all about the blue-white screening and its concept. Thank you for watching, and subscribe for more updates.